Good morning, all of you. Today we discuss the important cucurbits crop, cucumber. Budding of cucumber. We start from its botanical description. Its botanical name Cucumis sativus, family Cucurbidaceae, chromosome number 2n is equal to 14, origin northern India. Floral biology of cucumber. See the floral biology. First start from inflorescences. Inflorescence is cymose, cucumber is monoecious. We already discussed what is monoecious. Then flowers are bracteate, pedicellate, unisexual, actinomorphic, pentamerous, and epigynous. Anthesis means opening of flower. The whole development process from the initial bud stage to the stage when the flower is detached from the pedicel was divided into 8 stages such that is first opening of flower and closing of the male flower are immediately influenced by the sunrise and sunset, that is by the light and the time of the day effect. Flower are bracteate, pedicellate, unisexual, acetomorphic, pen, pentamerous, and epigynous. See the image of male flower. This is the male flower and this is a female flower. This is the difference between male and female flower. At the time of flower dissection, it is very important to dissect or to identify which is male flower and which is female flower. Then dissect out their part because it is a cucumber is a monoecious. Okay. Then anthesis. Anther-deviations between temperature ranges 20 to 20.5 to 21.5 degrees Celsius. Pollen fertility was considerable up to noon, and by afternoon, fertility was greatly reduced and was negligible by the evening. Means at the pollen fertility are increases at the time of sunrise and it is decreases at the time of evening. Means light effect is very important for the fertilization and anthesis of the cucumber flower. The stigma receptivity is for short duration and pollination should be carried out within 2 hours after anthesis. Means just opening of flower in between 2 hours the stigma is very receptive for the pollen for the fertilization. That's why the stigma receptivity period is a 2 hours after just opening of the flower or just after the anthesis. Rise in temperature causes early drying of stigmatic secretion. Means after morning the temperature increases. That's why effect the drying of stigmatic fluid in the stigma portion and pollen does not receive by the stigma. That's why the pollination failure. So we go for the pollination and fertilization at the morning period, just anthesis means just opening of the flower. Next, breeding objective of cucumber. First, reduce cucumber of nodes to first female flowers. Mean reduction in length of nodes in between the length of the nodes reduction, and then also number of female flowers reduction. Okay. Then bushy plant type, that is dwarf cultivars. Okay, means in between node portion uh, the length is very short. That's why the bushy plant. If we go the number of female flowers in one plant, the size of the cucumber is reduced. Ultimately, quality of the cucumber is reduced. That's why we reduce the number of nodes of female flower at initial stage. Then moderately bitter, lightly bitter, less mature seed, self-staking habit, that is dwarf character, high female male sex ratio as compared to male, female flower are more but not excess. Then more flesh thickness, desirable fresh color, attractive green or dark green fruits with smooth surface. Then next, Fruit shape, uniform, long cylindrical shape. Uniformity of fruit, size 10 to 15 centimeter long. Less seedlessness, reduce or resistance to parasitic or parasitic and non-parasitic diseases. Less or negligible curbitacin content, that is bitterness in cucumber, that is curbitacin. Then resistant to diseases like powdery mildew, downy mildew, and mosaic viruses like. Okay, and resistant to important pests like red pumpkin beetle and fruit fly. These are the breeding objective of the cucumber crop. The next important point, that is methods and achievements of cucumber go for the breeding. First method is introduction. By using the introduction method in cucumber, we develop the number of cultivars like Japanese long green, straight 8 and poinsettia high yielding variety. There is three variety, that is Japanese long green, straight 8 and poinsettia. These are the important cultivars which is developed by introduction method. It is very high yielding varieties. Next important method, that is mass selection. Cucumber lines with desirable characters like bushy habit, spineless, and lack of bitterness have been obtained by selection method, in which Sheetal is a very important or famous variety from the selection, from the mass selection. Then Kira 19, then Kira 75. These are the important cultivars by developed by mass selection method. Next, last important breeding method, that is hybridization, in which we cross the two cultivars which resistance or good characters according to breeder point of view and develop new important variety. Like Hemangi, it is developed by our MP K V Rauri University, that is Himangi variety. It is cross between Poinsettia into Kalyanpur Agiti. Next one, Phule Shubhangi, also same. Then heterosis breeding, that is Punakira into Japanese green long, then Chowbatia local into Solan local, then Kalyanpur Agati into Panvel. These are the important cultivars and cross or heterosis in cucumber crop. This is the method of breeding in cucumber crop, that is introduction, selection, in which mass selection, hybridization and heterosis. These are the important breeding method and achievements we discuss according to the methods wise. Okay, so thank you all of you for watching this video.